Our hearing will now come to order. I want to welcome everyone to today's hearing on biological research for energy and medical applications at the Department of Energy Office of Science. Our hearing today will explore the Office of Science's biological research programs and how they fit in with our broader federal research infrastructure for energy, environmental, and medical applications. The Department of Energy's role in examining biological processes is not always well understood, nor is it appreciated always, but it dates back to 1946. At that time, of course, we needed to learn more about the effects that radiation could have on people from the use of either atomic weapons or nuclear power. This required bringing together the best and brightest researchers from both physical and medical sciences to study the issue. Over the years, DOE developed unique engineering capabilities within its national laboratories that allowed the department to quickly catalog the building blocks of living organisms. These technologies are what enabled the Human Genome Project to be considered by scientists at DOE and NIH in the late 1980s, and then successfully completed that project on budget and ahead of schedule by 2003. Today, the Office of Science focuses these capabilities on developing next-generation biofuels, finding new ways to sequester carbon, and on cleaning up the legacy waste from our nuclear weapons complex. In addition, DOE's nuclear physics program has recently shouldered the responsibility of providing critical non-commercial isotopes for cancer treatments as well as other research applications. I look forward to learning more about the progress DOE is making in working with NIH and other agencies to meet the science and medical community's needs. And with that, I'd like to thank this excellent panel of witnesses for appearing and yield to our distinguished ranking member, Mr. Inglis.
Thank you, Mr. Chairman, and thank you for holding this hearing. Today we're going to find out about the complexity of the Department of Energy's Office of Science. Biology isn't the first thing that comes to mind when we think of critical research gaps in developing new energy technologies. But the Biological Environmental Research Program at the Office of Science is currently advancing biofuel development, helping us better understand the impacts of climate change on our environment and improving medical technologies. Research in 1949 about the health impacts of radiation exposure has evolved into dramatic advancements in genetics, radiology, and nuclear medicine. One of the most notable achievements of biological research at DOE is certainly the Human Genome Project. In coordination with NIH, the project resolved the complex human DNA sequence in 13 short years. With a diversity of efforts at DOE, I'm looking forward to hearing about other potential breakthroughs from our witnesses, particularly in the area of biofuels. I should also admit to a parochial interest in the Biological Environmental Research Program. Clemson University in the upstate of South Carolina has a remarkable research program in the College of Agriculture, Forestry, and Life Sciences. Researchers there do considerable amount of work on the genomics and development of biofuel crops, and have collaborated with DOE on several such projects previously, as you point out in your testimony, Dr. Keasling. Again, I'm very much looking forward to the testimony of our witnesses. While much of the work in the Biological Environmental Research Program seems only loosely related to the overall mission of DOE, they're working on exciting progress in a variety of energy and medical initiatives. Thank you, Mr. Chairman, for holding the hearing, and look forward to hearing the witnesses.
Thank you, Mr. Inglis. It's my pleasure now to introduce our distinguished witnesses at this time. Dr. Anna Palmisano is the Director of the Office of Biological and Environmental Research at DOE. Dr. J. Keasling is the Acting Deputy Director of Lawrence Berkeley National Laboratory and Chief Executive Officer of the Joint BioEnergy Institute at DOE. Dr. Allison Campbell, we are proud to say, is the Director of the W.R. Wiley Environmental Molecular Sciences Laboratory at Pacific Northwest National Laboratory, near and dear to my heart. Dr. Ari Patrinos is the President of Synthetic Genomics Incorporated. Dr. Jehanne Gillo is the Director of the facility. Did I say that right? Gillo. Gillo. The way it's spelled, not the way my staff wrote it. They wrote G-E-L-O, soft G. That would be Gillo. Are you sure you're right? Okay, we'll go with Gillo if you say so. After all, you are the Director of Facilities and Project Management Division in the Office of Nuclear Physics at DOE. As our witnesses should know, you'll have 5 minutes for your spoken testimony. Your written testimony will be included in the record. When you've completed your spoken testimony, we'll begin with questions. Each member will have 5 minutes to question. Again, I just want to apologize. We normally have a pretty packed house on this panel, but with the early dismissal today, folks are racing home to their districts. Some have said they'll try to make it. They also have, believe it or not, many other hearings conflicting with this. But we have an incredibly distinguished panel. We look forward very much to learning your input, and please, we'll ask Dr. Palmisano to begin, please.
Mr. Chairman, Ranking Member Inglis, and members of the committee, I appreciate the opportunity to appear before you today to discuss the Biological and Environmental Research Program in the Department of Energy's Office of Science. I'm the program director. Biological and Environmental Research, known as BER, supports innovative and transformational science to provide a fundamental understanding of biological, climate, and environmental systems. Through our research programs and our scientific facilities, we support a wide range of disciplines to engage a broad scientific community, using peer review to ensure scientific excellence. The BER program addresses three major scientific challenges. The first challenge is to explore the frontiers of genome-enabled biology. BER supports research that uncovers nature's secrets to harness the catalytic power and biomass of microbes and plants for bioenergy, the carbon cycle, and bioremediation. Starting with an organism's DNA, BER-funded scientists seek to understand whole biological systems as they interact with their environments. The second challenge is to discover the physical, chemical, and biological drivers of climate change. BER plays a vital role in the U.S. Global Climate Change Research Program by improving predictive climate models and by addressing some of the key uncertainties, such as clouds and aerosols and the carbon cycle. The third challenge is to seek the scientific basis for environmental sustainability and stewardship. The Earth's subsurface is a new frontier for discovering novel microbes and understanding geochemical and hydrological processes that affect the fate and transport of environmental contaminants. BER supports three world-leading scientific facilities that benefit a broad community of scientists. The Joint Genome Institute provides state-of-the-art genome sequencing and bioinformatic analysis for microbes and plants of energy and environmental significance. To date, the Joint Genome Institute has sequenced over 500 microbes in microbial communities, as well as 25 plants. The Environmental Molecular Sciences Laboratory provides novel experimental and computational tools for molecular-level studies of the environment. The Atmospheric Radiation Measurement Climate Research Facility provides unmatched level of observations and measurements of clouds and aerosols for climate researchers. BER-supported biological research has a long history of major contributions to the DOE mission and national needs through discovery, science, and innovation. Today, BER supports genome-enabled research to understand biological systems ranging from single microbes to microbial communities to plants. Our ultimate goal is to predict, manage, and control biological systems to support mission needs in bioenergy production, climate change, and environmental stewardship and sustainability. In September 2007, 3 DOE bioenergy research centers were launched to provide transformational science to overcome the most difficult scientific and technological barriers to the production of biofuels. Scientists are using systems biology to discover and optimize enzymes, microbes, and plants that will lead to new approaches to cellulosic biofuels. BER is deeply committed to coordination with the DOE's technology offices to facilitate a smooth transition of knowledge to application. Successful mechanisms for coordination include participation in joint reviews, site visits, science team meetings, and strategic planning. BER-supported research provides the fundamental knowledge of microbes and plants needed by the DOE's Office of Energy Efficiency and Renewable Energy for the successful development and deployment of new bioenergy crops for sustainable biofuel production. BER research on the fate and transport of contaminants in the subsurface environment provides knowledge for DOE's Office of Environmental Management to develop new strategies for stewardship and remediation of contaminants, and for DOE's Office of Legacy Management to develop tools to monitor contaminants at cleanup sites. Looking to the future, BER will strive to continue to advance the nation's biologic, climate, and environmental science through leading-edge programs that meet DOE needs. Thank you, Mr. Chairman, for providing this opportunity to discuss biological and environmental research program at the DOE's Office of Science. This concludes my testimony, and I'd be pleased to answer any questions you may have.
Mr. Chairman, Ranking Member Inglis, and distinguished members of the committee, thank you for the opportunity to testify today and for your strong support for science. My name is Jay Keasling. I'm the CEO of the Joint BioEnergy Institute, acting deputy director of the Lawrence Berkeley National Laboratory, and a professor of biochemical engineering at the University of California, Berkeley. I'm honored to testify before you today about the bioenergy research centers, which are advancing the science and technological development of cellulosic-based biofuels. From biofuels to cost-efficient remediation of toxic environments to changing the way we understand and predict global impacts of climate change, BER serves an irreplaceable role in the federal research enterprise. At the core of BER's strengths are its unique facilities and world-leading scientists. Since spearheading the Human Genome Project in the 1980s, BER has led advancements in modern systems biology that today enable the cutting-edge research into sustainable energy alternatives. Upon this foundation, BER established 3 centers to research and develop cellulosic-based biofuels. These are the bioenergy research centers, which today are up and running and making great progress. JBay's sisters are the DOE Great Lakes Bioenergy Research Center at the University of Wisconsin, led by Tim Donahue, and the DOE Bioenergy Center at DOE's Oak Ridge National Laboratory, led by Martin Keller. JBay is led by Lawrence Berkeley National Laboratory in partnership with Sandia Labs, UC Berkeley, UC Davis, Lawrence Livermore National Lab, and the Carnegie Institution for Science. The mission of the BRCs may be simply stated to advance the development of cellulosic biofuels. However, the challenge is grand. Unlocking the energy potential in the sugars of cellulose require a lot of basic research and technology development. The BRCs are ideally suited to make rapid progress toward this goal. Although unique in many ways, each of the BRCs has pulled together the best of the national laboratories, academics, and the private sector to build a new model for interdisciplinary research. Working collaboratively, the 3 BRCs have the potential to provide a better investment for the federal dollar than a single large center and may serve as a good model for similar energy research challenges. Let me take a moment to describe JBay in more detail. JBay is dynamically organized with scientific teams working together in a single location under one roof to enable researchers to share ideas and address problems at a systems-wide level. Researchers don't have to wait for the weekly conference call or the annual retreat to connect. It happens all of the time. Organized like a startup, JBay is designed to be nimble and flexible, able to focus and refocus resources quickly, not the typical research model. Unproductive research avenues are quickly redirected. Ideas that show the most promise are invested in aggressively. JBay researchers are focusing on developing next-generation biofuels that are compatible with existing infrastructure and utilize feedstocks more efficiently. Taking a whole systems approach to this objective ensures that our research is applicable on large scales. 4 independent areas are investigated: developing new bioenergy crops, enhancing biomass deconstruction, producing new biofuels through synthetic biology, creating technologies that advance biofuel research. The magic of this approach is that advancements in any of the 4 areas can be shared with and employed by areas, by other BRCs, and by industry. The exciting research includes searching for new ways, including novel and better enzymes to break down lignocellulose, the tough matrix of fibers that hold plant material together. An answer may be found in microbial communities in Puerto Rican rainforest soils that boast some of the planet's highest rates of biomass degradation. JBay researchers are analyzing these organisms to find potential solutions. On the fuel production side, using synthetic biology, JBay researchers have re-engineered the microbes of E. coli and yeast to produce advanced drop-in fuels that perform better than ethanol. Basically, these tiny microbes can become biofuel refineries. My personal area of research is in synthetic biology. In addition to biofuels, this exciting field offers great promise for bio-based chemical and medical products. One of the most important applications of synthetic biology has been re-engineering organisms to produce the anti-malarial drug artemisinin. There are currently 300 to 500 million cases of malaria at any one time, with 1 to 3 million people dying of the disease each year, and 90% are children under the age of 5. And while quinine-based drugs are no longer effective, plant-derived artemisinin combination therapies are highly effective but cost-prohibitive for the world. To decrease its cost, we engineered a microbe to produce a precursor to the drug by transferring the genes from plants to the microorganism. The process has been licensed by Sanofi Aventis, which will scale the process and produce the drug within the next 2 years, providing it at cost to the developing world. Luckily, the precursor to the chemical artemisinin is a hydrocarbon, a fundamental building block of fuel. We are now re-engineering those same microbes to produce drop-in biofuels. The artemisinin project required $25 million in funding and 150 person-years to complete, in part because the engineering of biology is so incredibly time-consuming. Through synthetic biology, we hope to make the engineering of biology more predictable and easier, thereby reducing its cost to develop biofuels and other useful products, from chemicals to medicine to consumer and commercial products. Limiting BER research to just fuels would be a mistake and a lost opportunity. Indeed, BER can take an important and leading role in the development of this transformative field of synthetic biology. Thank you again for holding this important hearing and for inviting me to participate, and I'd be happy to answer any questions.
Thank you, Chairman Baird, Ranking Member Inglis, members of the committee, for the opportunity to appear before you today. I'm the director of EMSL, the Environmental Molecular Sciences Laboratory, a BER-funded national scientific user facility. EMSL's mission is to provide researchers worldwide with integrated computational and experimental capabilities to advance scientific discovery and provide technological innovation in the environmental molecular sciences in support of DOE and the nation's needs. It is unique in that it offers users under one roof a problem-solving environment that integrates these capabilities with staff expertise that enable the highest impact science possible. Capabilities include high-performance computing tools, ultra-high-resolution microscopes, world-leading magnetic resonance spectrometers, think of it as an MRI for molecules, and mass spectrometers. Within the Office of Science, BER supports, sponsors, and advances world-leading biological and environmental research programs and operates scientific user facilities that drives fundamental scientific discoveries to meet its mission priorities. In addition to DOE's Office of Science, the National Science Foundation and the National Institutes of Health also fund programs in biology and medical research. Many scientists funded by these three agencies perform their research at DOE-sponsored national scientific user facilities such as EMSL. A few examples of highlights in biological area include researchers from the Washington University at St. Louis who have recently discovered a novel cluster of genes that encode proteins essential for photosynthesis. This is the process by which plants convert light into energy. Understanding this process and how nature converts light into energy is a reaction important in the development of new clean fuels. Another example is researchers from Oregon State University and the University of California, as well as at PNNL, for the first time measured protein complements of microbial communities in the Sargasso Sea. Insights afforded by this research is important because bacteria such as these heavily influence biogeochemical cycles, affecting the concentrations of elements such as carbon, and therefore the greenhouse gas carbon dioxide in the Earth's air, water, and soil. Finally, an international team from the Erasmus Research Center at Rotterdam have identified 55 different proteins that vary in amounts between patients who are responsive to a certain breast cancer therapy and those who are not. This discovery can potentially lead to new biomarkers for the efficacy of new therapies and drugs. BER continues to make significant investments in EMSL to keep the user facility unique and state-of-the-art, such as the recent investment of $60 million of Recovery Act funds to enable our planned investments in recapitalization. We are also collaborating with the National High Field Magnet Laboratory at Florida State University, as well as an institute in the Netherlands, to develop the world's highest field mass spectrometer. This high field magnet would make what today is impossible possible through increases in dynamic range, sensitivity, and resolution. New knowledge garnered from this instrument could enable biofuel development and foster better informed technology and policy decisions affecting bioremediation, waste processing, energy production, and associated health impacts. Of course, EMSL would not exist without our user base. During our 12 years of operation, we've hosted more than 10,000 scientists from all 50 states, including all the states represented by this committee, in over 60 countries. Nearly half of our users come from university systems. 40 come from other national labs and other government labs, and a small portion come from the industrial sector. Nearly 45% of EMSL users are funded by DOE, with one-third of those being funded by the Office of Biological and Environmental Research, and another 25% are funded by NIH and NSF. The remaining balance being funded by various associated agencies across the government sector. User productivity has been excellent. Over the last 12 years, EMSL-based research and discoveries have been the subject of more than 1,000 peer-reviewed papers and journals, and featured on more than 30 journal covers. To summarize, in partnership with BER, EMSL will continue to provide these world-class scientific resources and scientific expertise to the scientific community worldwide, with integrated capabilities to achieve the highest impact science possible in support of the needs of DOE and the nation. Thank you, Mr. Chairman, for the opportunity to discuss EMSL and DOE's biological programs with you. As we both call Washington our home, I'd like to invite you at your convenience out to the laboratory to take a look yourself, and I'd be pleased to answer any questions the committee might have.
Thank you, Mr. Chairman, Ranking Member Inglis, and Mr. Ehlers. I am honored to be asked to speak about BER and about my company, Synthetic Genomics Incorporated. I'm also pleased to see that my colleagues on the table also still recognize me and remember me. The common theme between BER and my company, Synthetic Genomics Incorporated, is in fact genomics, which you've heard so much already. SGI, the company, was created by genomics pioneer Craig Venter in the summer of 2005 to drive commercial solutions using genomics, starting with energy, but eventually we expect to move into things such as vaccines, clean water, and many other applications. We are currently partnering with industry giants like BP to enhance hydrocarbon recovery, subsurface hydrocarbon recovery, with a Malaysian company, Genting Asiatic, to sequence the genomes of jatropha and oil palm, and the microbial communities residing in the rhizosphere to include things such as yields. And very recently, we also announced an alliance with Exxon to exploit algae-produced biofuels. The genomics revolution, as you correctly have stated, started really with the Human Genome Project that was launched by the BER program back in 1986 by one of my predecessors, Charles DeLisi. Through the genomics program, through the Human Genome Project, we had developed many high-throughput technologies for sequencing, assembly, and informatics. Many of those technologies actually developed by Craig Venter himself. Over the years, there have been very many successful partnerships between BER and Craig Venter, and in fact, one of them continues to the day. As you've heard already, synthetic biology, synthetic genomics, genome engineering are all new fields that have been essentially been launched by genomics and promise disruptive technologies with myriad applications beyond energy in medicine and in other industrial processes. I am very proud of my many-year association with the BER program and for the contributions the program has made since its inception. I will always remember the age of the program because it's the same age that I am, 62 years, and the contributions it has made in radiation biology, in nuclear medicine, climate change, bioremediation, genomics, structural biology, the list goes on. BER has always invested in high-risk and high-payoff research and leveraged the physical and the computational sciences that reside within the Office of Science, that unique position that BER enjoys. BER, therefore, should not be like NIH or NSF. It should retain its own DNA, so to speak, because diversity is really the strength of the American scientific enterprise. And I mean diversity of performers, diversity of scientific approaches, and yes, diversity of funding sources. A good idea that gets shut out by an agency that dominates a field needs another chance to be shopped around. If it wasn't for the BER program, the Human Genome Program would probably be much delayed, and probably we would still be sequencing it. When I was in DOE, I suffered in the last few years by much questioning about why DOE should be doing biology. Questions came from the Secretary's office, from the OMB, and also from your sister Committee of Energy and Commerce. There was also an attempt to raid BER to provide funding for the newly founded Department of Homeland Security. I am hopeful that these dark days are over and my successor will not have to suffer what I suffered back in those days. BER is extremely important for the DOE missions, especially those involved clean energy, as you have heard. Carbon capture and sequestration will not be possible without a biological solution, and also bioremediation of the legacy of the Cold War. Also, BER has important scientific user facilities, like you've heard, the Environmental Molecular Sciences Laboratory, the Joint Genome Institute, and the facilities and stations it nurtures at the light sources and the neutron sources at the national labs. My suggestions for continuing the successful tradition of BER is to push the high-risk, high-payoff envelope. Too much is at stake, especially for climate, not to do that. Continue to exploit the physical and computational sciences that reside in the Office of Science. There are still many new tools and methodologies that BER can steal shamelessly in order to serve the biology. Also, nurture public-private partnerships. I particularly appreciate that now being in the private sector. There are obstacles like intellectual property, but these obstacles should not stand in the way of making them successful. And build the scientific infrastructure for synthetic biology, including looking at the ethical, legal, and social implications of this new disruptive technology. Finally, I must say that the BER stewardship role for genomic science, Mr. Chairman, Mr. Inglis, Mr. Ehlers, needs to be affirmed, needs to be strengthened, and generously funded if we are to successfully confront the great challenges of our times. Thank you very much for the opportunity to testify, and I would be delighted to answer any questions you may have.
Thank you, Mr. Chairman, Ranking Member Inglis, and members of the committee for the opportunity to appear before you to provide testimony on the DOE Office of Science's Isotope Development and Production for Research and Applications program within the Office of Nuclear Physics. The isotope program was transferred from the Office of Nuclear Energy to the Office of Nuclear Physics in March 2009, and specifically to the Nuclear Physics Facilities and Project Management Division. I've served as the director of the division since 2004, and I'm pleased to share with you my perspectives on the DOE isotope program. The Office of Science recognizes that isotopes are high-priority commodities of strategic importance for the nation and essential for energy, medicine, national security, and scientific research. And a goal of the program is to make critical isotopes more readily available to meet domestic needs. The expertise of the nuclear science community in operating accelerator facilities and developing instrumentation and accelerator technology for a broad suite of applications complement the expertise of the isotope production community. And the synergies between the two communities will lead to an overall improvement in the productivity of the isotope program. The isotope program produces isotopes only where there is no U.S. private sector capability or other production capacity is insufficient to meet U.S. needs. Isotope production for commercial distribution and application is done on a full cost recovery basis. Isotopes are needed for a broad range of basic research, biomedical, homeland security, and industrial applications that benefit society every day. For example, americium-241 for smoke detectors, helium-3 for neutron detectors, nickel-63 for explosive detection, strontium-82 for heart imaging, californium-252 for oil exploration. Isotopes have had a profound impact on daily life, including reduced healthcare costs, improved ability of physicians to diagnose illnesses, and advances in agriculture and basic physics research and the security of the nation. The isotope program supports both production capabilities at a suite of facilities as well as the research and development efforts associated with improving and developing isotope production and processing techniques. As a service, the isotope program also sells and distributes other isotope products that it does not directly produce. Examples are helium-3, lithium-6 that are produced by the DOE National Nuclear Security Administration, or NNSA. The isotope program does not produce special nuclear material or sell highly enriched uranium. The isotope program is not responsible for the production of molybdenum-99, a medical isotope which currently is in short supply. The DOE NNSA is responsible in the long term for establishing a diverse domestic supply of molybdenum-99 without using highly enriched uranium. The Office of Nuclear Physics has taken several actions to improve communication amongst isotope stakeholders. A workshop was organized last summer to bring together university, laboratory, federal, and commercial isotope producers and users to discuss issues related to isotope production and identify isotopes in short supply. The Office of Nuclear Physics has specifically engaged federal agencies in discussions regarding agency needs and concerns on isotope production, including the National Institutes of Health, the Department of Homeland Security, and NNSA. The program is in the process of increasing the suite of production facilities with consideration given to the capabilities at universities, commercial entities, and other government facilities. The research component of the isotope program is being strengthened within nuclear physics. Research and development efforts associated with improving the effectiveness or creating altogether new approaches to isotope production are being pursued. Research isotopes will be produced more reliably and at more affordable prices. In 2008, the Nuclear Science Advisory Committee our federally chartered advisory committee was charged by the Office of Nuclear Physics to develop a prioritized list of research topics across a wide range of scientific disciplines that used isotopes. The committee was also asked to develop a long-range strategic plan for future production of stable and radioactive isotopes. The Office of Nuclear Physics is committed to increasing availability of isotopes in short supply, providing isotopes reliably and at more affordable prices to researchers, and supporting research activities that develop more cost-effective and novel isotope production techniques. NP is using merit peer review and priority setting mechanism to optimize the productivity of the isotope program within available resources. Thank you, Mr. Chairman and members of the committee, for providing the opportunity to discuss the isotope program, and I'm happy to answer any questions that you may have.
I thank the panel for most informative testimony. You're doing great work. I'll recognize myself for 5 minutes, and then we'll proceed with my colleagues. Help me understand, Dr. Patrinos, it's good to see you again. You were here previously on some of those prior hearings. I remember them. Walk us through, though, a little bit about the interface between, let's say, NSF, DOE, now ARPA-E, and how research is, how do you make sure that we're not all doing the same thing, or we're not neglecting the gee whiz discovery that's going to solve the problems? How do you sort that out? How do you coordinate with those other agencies, or how do you differ in some ways?
Thank you for your question, Chairman Baird. All of our program managers are actively engaged in interagency working groups under the aegis of the National Science and Technology Council and the Office of Science and Technology Policy of the President. And through these relationships, we build joint programs. We ensure that our programs are synergistic and complementary, and then we minimize the amount of overlap that exists. And I'd be happy to give examples if you'd like.
So if NSF is focusing its funding, grant-related funding in some area, you would say, okay, you've got that covered. We're going to look in a different way?
Yes, sir. That's exactly correct. And I can give examples of where, for example, we've partnered with other agencies such as the U.S. Department of Agriculture and BioEnergy, where we realized that we had both had interests in biofuels, but we had complementary expertise. So we launched a program on plant genomics for bioenergy feedstocks, and that took advantage of the USDA's expertise in traditional plant breeding and cultivation of bioenergy crops, and DOE's expertise in genomics. So there are many similar examples of that with the National Science Foundation, NIH, and other agencies.
That's very helpful. The argument, of course, a while back was, well, to what extent is this duplicative? Do we have multiple bureaucracies trying to do the same thing? Can we not save money, et cetera? And I think there's a great deal of merit to the cross-disciplinary synergies that you describe. And though I think unexpected, when a layman looks at DOE to see your operation, it's unexpected. The proof's in the pudding to some extent, and you've done some remarkable things, and I think that's commendable. Dr. Keasling, you sort of raised an intriguing question that I want to follow up on a little bit. Two things. One, you spoke about limiting research to just fuels would be a mistake and a lost opportunity. Are you feeling like it is limited, or you're concerned about the potential that it would be limited?
I'm more concerned about the potential that it would be limited. I think now that BER has gone down this path of biofuels from biomass, which is a great thing for it to be doing, we could potentially source all of our petroleum-based, all the chemicals that we now source from petroleum, but from biomass or from sugar. And so there's huge potential there. And the research would be directly complementary to what's already being done in BER. So it's a potential growth area.
I'm not sure I understand. You're saying because it's a growth area, that would rule out some of the other research, or?
No, I'm saying that it's an additional area of research that could be done by BER.
Okay. Related, and actually the next area in your testimony, you had talked about, and you worded it artfully, so I don't want to try to get you in trouble with if this would do so, but it was an important issue that, if addressed effectively, could improve the department's ability to develop solutions. And the issue there seemed to be that the energy research seems somewhat disconnected from the basic sciences. Can you elaborate on that?
In fact, we are using a lot of the basic science research that BER has developed over the years. So a lot of the basic research that's going on in their GTL programs in the Joint Genome Institute, all of this is extremely important to the work that we're doing on converting biomass to biofuels. So in fact, that core basic research is so important. And the work we're doing, while it has an important goal of breaking down these barriers of biomass to biofuels, it's still fundamental research.
Okay. Are there artificial constraints, and this is for anybody, where you feel that you have the expertise and knowledge to make major contributions in an area that's consistent with your mission, but we've somehow statutorily or historically constrained anyone here, or are you feeling you've got some of the leeway to do what you need to do?
From my own perspective, the research we're doing is something that I would want to do anyway.
Yeah.
It is important, though, for us to make a connection to energy, we feel, as we're proposing this research. So,
the connection's great, but at the same time, you gave a point about artemisinin, and
right.
That's a big deal. And if if you find that thread, you ought to be able to pursue it.
That's right. That's right. And artemisinin is a unique case because it's a hydrocarbon, so it's not too far a stretch from biodiesel.
Great. Proud to recognize Mr. Inglis for 5 minutes.
Thanks, Mr. Chairman. So Dr. Keasling said something interesting that I'd like to compare with what Dr. Patrinos said about funding. Dr. Keasling, you said it's important to figure out which research paths are dead ends and cut them off quickly, which makes a lot of sense to me. And but then it's also true, Dr. Patrinos says you want diversity of funding sources, and I guess that's in order to develop kind of paths that maybe aren't too clear. So how do you work that out? I think it's an impossible question, actually.
That's an excellent question. And I was speaking more about how JBay manages its research portfolio. And one of the things that we wanted to do when we designed JBay is be able to go down a path that looked like it was going to be productive as quickly as possible and see if it's going to be a productive angle of research. And if not, then we redirect those research funds to another area, an alternative area that looks like it's going to be productive, so that researchers don't spend all of their time going down a particular path that will eventually be non-productive, but they're doing so because they don't have any flexibility. So we've built in this flexibility in JBay from the top that allows us to really focus on an important aspect, and if it doesn't work, go to the next aspect of the problem.
Which is a difficult thing to do, right? Because people say they've got their PhD in a particular area. That means that their sort of meal ticket is punched by that area. And so if you find out that that's not productive, they just lost their meal ticket, right? And so it's maybe there's a way that you do that. It's a tough management challenge, I take it.
It is a tough management challenge, but because there are so many important problems. If we take, say, the plant area, for instance, we have researchers in J-Bay who specialize in plant genetic engineering and understanding how cellulose is made in plants. So they could be looking at one particular aspect of how cellulose is made, and maybe that won't work, or it doesn't look like we can increase the cellulose level. So they'll turn their attention to a different way in those same plants, or still using plants to increase the cellulose level. So it's a little more subtle than completely cutting off an entire research area. And we do, to the extent we can, try to preserve people's meal tickets.
Right. Well, Dr. Patrinos, anything to add about that, about how you balance that?
Well, I'd like to say that basic research is fundamentally a messy housewife. And the tendency is always by, especially newcomers in political positions, to tidy up research, to look for redundancies and remove those because, that way we save money and so on. It turns out that the more you try to tidy up, the more you restrain the research. There has to be redundancy because there has to be competition and there has to be diversity in approaches. I mean, if I was still in DOE, I would have said the same thing my colleague Anna Palmisano said, that we did a lot of collaborations when I was in DOE across agencies, and I think my record speaks for itself in terms of the partnership with NIH, with NSF, and other agencies and so on. But I don't hide the fact that there was also competition. We have different attitudes, different approaches, and we represented different cultures. And even though there was occasional arguments, sometimes pretty violent, I would say, the net result was always very, very positive. It was the give and take of competition, the give and take of having different points of view that were brought to the table, and the ultimate result in the conduct and execution and management of research is so far better than anywhere else in the world because of that perceived untidiness.
Yeah. Dr. Palmisano, do you want to add anything to that, or how your approach may differ or be consistent?
Yes, Congressman Inglis, I agree with everything that Dr. Patrinos said, and I think I describe it that we challenge one another in a very positive way to provide the best we can for the American public. And I think that there is a very good balance and dynamic among the different agencies pursuing science for that reason.
Yeah. Thank you, Mr. Chairman.
Thank you. Dr. Inglis, or Dr. Ehlers.
Thank you. And just a quick side note a minute. I agree, Dr. Patrinos said basic research is messy, at least the way I did it was. But I'm puzzled why you blamed a housewife instead of a house husband. I find house husbands are much messier than housewives. Yeah, it's okay. Don't take me seriously. Dr. Gillo, I must admit I'm suffering from some sleep deprivation, but I don't quite see how the isotope production relates to what we're doing. And first of all, what isotopes are you talking about producing, and how does that relate to the energy generation issue? Could you run through that again, please?
So the isotope program that was just transferred to the Office of Nuclear Physics has two components: to produce isotopes for basic research and also for a broad suite of applications. And so we operate accelerator facilities and also make use of other facilities domestically, reactors, accelerators, to produce these isotopes and to distribute them as a service to the nation. Okay. They're used for energy reasons. They're used by the BER program and
okay,
other.
That part I understand, but how does it relate to the cellulosic issue and the energy production issue? Are these used as tracers in some of the experiments?
They can be used as tracers. Yes, they are used. Are
these by and large radioactive isotopes?
They are radioactive isotopes and they are stable isotopes. The stable isotopes, we have an inventory that we distribute, and the radioactive isotopes we produce. And so the BER program are users. The NP program, we are the producers of the isotopes. And yes, they're used as radiological tracers in plant studies and other life sciences.
Okay. Now, the non-radioactive ones, you trace them with mass spectrometry and so forth? Or?
Yes, they're used primarily. One of the ways that they're used is for nutritional studies, since they are non-radioactive. And so that's one of the most popular bone studies, retention in bone growth, osteoporosis studies.
Okay. Thank you. I wonder if some of you could give me the broad perspective here. You know, everyone got excited about ethanol here a few years ago, and we passed some legislation which I thought was probably unnecessary and perhaps damaging. And I attribute that mostly to the farm lobby rather than the scientific community. And I think my impression has been borne out that it's not the best way to go. But when you, just picking up what you said, it seems to me you're still talking a lot about ethanol, but producing it with cellulosic material. Are you looking at other fuels? And what other fuels are you looking at?
Well, I can start, Mr. Ehlers. We believe in synthetic genomics that corn-based ethanol especially is a big mistake.
Yeah.
In some way, of course, we benefited because through that process we cut our teeth in the biofuels business. So at least there is some credit is due. But we need to move away from corn-based ethanol as far and as fast as possible. Any fuel that competes with food should really be not pursued. We should not pursue it. I also think that ethanol is not a very good fuel by itself. You know,
Right.
it doesn't mix with water. It's very corrosive. So it may have been a good start, but I think we need to be moving away from ethanol as well. So there are better quality fuels that we could pursue, even using cellulosic material, but also, as I mentioned in my introductory remarks, the use of algae to produce a variety of biofuels is perhaps the one that we think has the greatest promise.
And what type of biofuels would they produce?
Jet fuels is the fuel that we particularly are focusing on at this stage, but it doesn't necessarily have to be a fuel. The process can actually generate a crude that mimics in every way the crude that we remove from the ground, so we can insert it into the existing infrastructure for the production of a whole variety of fuels that we currently use. So that would be the ultimate holy grail of this enterprise.
I see. And what sort of chemicals do you pull out of this?
This is essentially different molecules of carbon, let's say starting from C12 all the way up to C20.
Oh, really? Hmm. And you get that large variety from the cellulosic material?
No, the one that I'm describing is using algae, carbon
sorry,
dioxide, and
okay.
sunlight.
Yeah. Okay. And you regard that as a very promising field at the moment?
We do indeed.
Yeah. Are there other promising fields that you're looking at?
This is not a renewable fuel per se, but we are looking to enhance the production of natural gas in existing coal beds, and thus avoid the need to extract the carbon and to burn it. So from a point of view of CO2 climate change impacts, it's a significant savings because burning methane is a lot cleaner than burning coal.
That's certainly true, but you still generate a lot of CO2 from
We
it.
generate CO2, but then I hark back to my first statement about using algae.
You're also
CO2
using carbon
The
there.
CO2 that's generated from the methane can then be recycled using the algae and sunlight.
Okay. I think my time has expired, so I better yield.
The great thing about Dr. Ehlers is he knows what he's talking about, so he can go on for a while, and we just watch and learn.
I'm just very good at pretending.
Give us some discussion of, Dr. Patrinos raised the issue of public-private partnerships, and one of the questions the public quite rightfully asks is, okay, so what's in it for them? Give us some examples, if you can, of Dr. Campbell, for example, and take just, for example, your work at EMSL. What are some examples of things that you think might have commercial applications, or where if I'm talking to John Q. Public about why do we do this research, what does the average guy get out of his or her investment in this endeavor? Give us some examples of that. Talk about how you would partner with a private company and what the implications are, et cetera, for that.
Sure. At EMSL, of course, since it's a national user facility, many of our users or some of our users come from industry, and they can either work with us in one of two ways. They can work in a proprietary manner where they pay the fee to operate and utilize the facility, in which case they would retain any intellectual property or knowledge that would result from that research. A more common way for industry even is to work in the open environment where they agree to publish. And many times they come with us in a lot of cases on the technology development side. So they may be interested in pushing a technology or an instrument forward. An example of that would be in our mass spectrometry area, where we're developing capability that would enable us to increase the sensitivity of certain commercially available mass spectrometers or the throughput of those mass spectrometers. We develop that, and then that would be commercially available and licensed out to these companies, for instance. And then the greater benefit of that is of course, the resulting science that these advancements have for the scientific community broadly. So if you can do things at higher resolution, at higher throughput, you can perhaps start to do clinical assays or clinical studies or more system-wide type studies. That gets published, it goes out to the broader scientific community in that regard. So you can have a direct line, or you can have a more indirect line where the knowledge base is created through these advancements.
So on the one hand, you have facilities that other people, maybe I'm a bright person, but I don't have the capital to build the kind of equipment you have, and maybe that nobody has that capital except the government.
Yeah.
And so the government's able to say, we'll make these resources available, and then people from private sector can contract with you to do that, right?
That's correct.
And at the same time, then you help refine the instrumentation that could be used by the private sector and
In partnership, oftentimes, with the private sector. So we have, for instance, a partnership with a company that builds probes that goes into these magnetic resonance spectrometers. They're interested in it because it can go into their product pipeline. We're interested in it because it can open up a whole new area of biological research, and that it'll allow you to look at proteins inside intact membranes. And so our users are now getting a new capability through this partnership with this company, and they're getting a new product pipeline. So it's a win-win, in my opinion.
do they pay, if I develop a product based on your work, is there a buy-dole kind of function? Do I pay back into the system, or how does that work?
Yeah, there's intellectual property rules that we follow, and it depends upon, of course, the type of the agreement or the relationship, where the government may take some ownership in the intellectual property, and then it can come back into the laboratory, or it might be an exclusive. It just really depends on the type of relationship.
Dr. Patrinos, now that you've made the jump to the dark side, I'm just teasing about that, but you've made this big move from director of a government program to the private sector. What insights have you gained about that? How do you, in terms of how we can do things better on the government side, or how private sector can interact better? What are your insights from that?
First of all, it may sound a little self-serving when I encourage you to foster more and more productive public-private partnerships. But I must say that even when I was in the Department of Energy, and specifically for the Human Genome Project, I advocated a very strong presence and involvement of the private sector. And in fact, I helped bring Celera Genomics to the table, and we successfully completed the program and avoided serious embarrassment at the time. But also created many partnerships that survive to the day and are extremely productive. So it isn't just self-serving. But nevertheless, my move to the private sector has very much reinforced my conviction that it really is the private sector that can translate successfully the wonderful discoveries that the programs like BER nurtures and translates them into real products and services. This is something that I've grown to appreciate a lot more than perhaps I theoretically or intellectually could accept in the past. It's already been mentioned, the kind of things that needed to be done. One of the areas that I feel needs to be strengthened further is creation of these scientific user facilities across a broader spectrum of the scientific disciplines. I think biology is tremendously benefiting from the light sources and the neutron sources and nuclear magnetic resonance facilities like EMSL, for example. But we need to do more for biology, because biology is the science of this century. And we need to provide the resources for all our scientists in both the public and the private domain. And they need these facilities, whether they are supercomputers for computational biology, or dedicated facilities for the production of proteins, for example, or doing the proteomics of looking at the entire protein components of an individual cell. These are capabilities that are in great demand, and if successfully put in place, will enable biology to very quickly deliver on the promises that it has, very rightfully so, of changing our lives, changing our industries, and solving many of our problems.
Dr. Palmisano, please share your insights on that as the current director.
You know, I think the future lies in our solving the problem of the vast amounts of data that are being generated through systems biology, our ability to manage those data, to mine them, to integrate them, to provide them and make sure they're accessible to a broad community of scientists, to assure their quality and standardization. And I think that's a major challenge that probably all of us at this table face. And we are, through the new sequencing, types of sequencing technologies, we're generating a huge amount of genome sequencing, proteomic data, metabolomic data. It goes on and on. Information about genetic networks, trying to combine that with computational models of biology. And, you know, I see that as really a need for the future.
So I don't really think the general public has a full appreciation, probably not this body itself, of this kind of model, of the basic science role, but not just the basic science in terms of the, okay, so the publication comes out and the data gets out, but the basic science in terms of the hardware, the physical infrastructure, the supercomputers, the light sources, the NMRs, et cetera, that the average guy just doesn't have access to. But really brilliant people can access it through your resources, and then you get a tremendous multiplier. We see it with nanotech as well, and some of the nanotech initiatives. And I think there's a whole host of, whether it's accelerators, that we really need to sort of highlight that. And this comes in the context of the sort of vitriolic debate now of, does government do anything well? Government does best what people can't necessarily do themselves, and this is something government does well. I don't think the average business is going to create, you know, light sources or accelerators or isotopes in some cases. Some do, obviously, they make a business model out of it, but in some cases we just have resources that allow us to do that, and DOE is an example of that. Mr. Inglis.
Thank you, Mr. Chairman. Following up on that, it's also true that private industry is the one that's going to implement the technology. So if, for example, Dr. Patrinos was talking about the use of carbon dioxide to grow algae, is that we need to do more of this research, I take it, in order to prepare for that. But there is a point at which you want it to tip over to have somebody actually building these things and using the CO2, right? How far away is that before we're really making use of the CO2 rather than wasting it?
Your microphone, Doctor.
It'll be several years before we can have large-scale recycling of CO2 through the method that I described, using algae and sunlight. Right. But nevertheless, the urgency is huge because of the problem of global climate change and the need to do something about carbon. Right. And therefore, if properly funded, both by the public and the private sector, I think we can see some of these advances happening faster, perhaps, than we may have assumed originally. This is the promise of biology. This is the promise of genomics. I make the parallel of, currently, the advances are on the surface. It used to be that you had to dig real deep to get a nugget of gold in the high-energy physics field. In biology, all you need to do is stoop down and you pick it up from the ground. This is the analogy that I have. Genomics has given us this power, has given us this fuel, and all we need to do at this stage is make sure the right resources are put in place so that we can fully capitalize on this capability.
So with limited resources, would you put your money on using the CO2 to grow algae, or would you put it on sequestration?
I would do both. I strongly believe that dealing with the climate change problem, we have a case of silver buckshot as opposed to a silver bullet.
Right.
We need to look at all forms of sequestration, just like we need to look at all forms of energy, renewable energy.
Is that because it's not possible to use the great volume of CO2? So basically, you've got to figure out some way to sequester it. Is that right? Or can you see a future where there's the use of CO2 is scalable to the point that you really could use, say, all coming out of a coal-fired electrical plant, for example, to create this biofuel?
Perhaps not all of it, but if we are successful in sequestering or recycling 50% of that, it's a long ways towards stabilizing the atmospheric concentration of CO2 if we combine that with aggressive use of renewable energy.
Interesting. Anybody else want to add to that?
I think that, when plants grow, they're scrubbing CO2 out of the atmosphere to make the biomass. And this is another way we can reduce carbon dioxide being put into the atmosphere by producing our fuels from that cellulosic biomass. And so just as algae do it and scavenge it to build algae, so do plants. And this is a great way to go for carbon-neutral biofuels.
Yeah, that's great. Yes.
So at this point in time, there's so much we need to learn about the carbon cycle. It's one of the greatest uncertainties in our climate model, some very fundamental information. And now we're starting to bring the tools of modern molecular biology and genomics to bear on the carbon cycle. And in doing so, we want to cast a wide net and use a number of different models, plant, microbial models, microbial communities.
Great. Thank you, Mr. Chairman.
Dr. Ehlers.
Is it fair to say that what you're doing with cellulosic materials, algae and so forth, is developing very, very sophisticated ways of using solar energy? Or is there more to it than that?
Well, nature's been doing this for a long time.
I know.
Right? So we are repurposing this source of biomass, or algae as it is, to produce biofuel. So it is a sophisticated form of capturing sunlight and carbon dioxide.
Yeah, because that's really your energy source.
That's correct.
And it's really the only relatively perpetual energy source we have.
That's right. The key, though, is to get them to produce the right fuels.
Yeah. Last round I had most of my questions for the end of the alphabet, or not quite the alphabet, but usually we go left to right, so I started the other way. But I want to give the three of you on that side a chance to respond to the questions I asked earlier. If you don't wish to, that's fine, but I just wanted to give you the opportunity.
Well, thank you, Congressman Ehlers, for that opportunity. One thing I'd like to comment on that you asked Dr. Gillo about was this, the use of radioisotopes, that we work very closely with Dr. Gillo and with our colleagues at NIH on to develop new types of radiochemistries that can be used as metabolic tracers for lots of different models, not just for humans, but for microbes and for plants. So we can start to understand, for example, carbon allocation in plants and microbes. So we've been able to take advantage of those opportunities that have been provided through our colleagues who are producing these radioisotopes.
Well, it's true. Radioisotopes are extremely convenient because they let their presence be known wherever they go, and with very specific signatures, so you can really track them very easily. Mass spectrometers work for those that aren't radioactive, but that's much more cumbersome. Dr. Patrinos, you said something like the next century is the century of biology, and I have to demur just a little bit on that because I remember, even though I wasn't alive then, but I've read the books, 1906, there were predictions that physics was essentially over. We had found everything that was to be discovered via physics, and the century turned out to be the century of physics. So I appreciate your comment. It made me think about it, but it tells me that some of our other branches of science better get busy too if they want to avoid the catastrophe of this being known as the century of biology. Now, of course, for biologists, that's a great thing if it happens. I really appreciate the insight you brought here. I've had lots of questions on this topic and just have not had the time to sit down and try and catch up on it. And you've done a very concise, a very good job of bringing me up to date. Thank you very much. I yield back.
Mr. Inglis had to race to catch a flight. I have just two quick more questions, and unless if any of you have to catch a flight, tell me. It occasionally happens that poor witnesses, we make a miss and they have to spend another day in this town. One of the questions, the Senate Energy and Water Appropriations Subcommittee has been looking at shifting nuclear medicine and medical applications in its jurisdiction to nuclear physics. Where do you think Dr. Gillo is an appropriate residence of this, if I'm not asking you to speak out of school? If I am, tell me. I'd rather not comment. But what's your expertise and insight into this?
I think the program is most optimized within BER. Within the nuclear physics program, the focus really is on the production of isotopes, not on the use of isotopes. And so it would be far more productive within the BER program.
Okay. It's not there now, though, right?
Yes, it is.
Okay. Oh, I'm sorry. Sorry. You were saying earlier it was within the nuclear physics program.
The isotope program is within nuclear physics. It is best to keep the medical applications
Gotcha.
program within BER.
Got you. Thank you. That's helpful. Dr. Campbell, talk to us a little bit about bioremediation, if you would. We've got the Hanford nuclear site upriver. Some of those isotopes make their way downriver. Talk to us a little bit about what's done to.
There's a lot of potential in bioremediation in that if you think about the isotopes that are of interest that have the potential to migrate out, for instance, to the Columbia, it's possible to transform those from mobile species, ones that migrate, to immobilized species, ones that don't migrate. That is often facilitated through microbial interactions. And that's a strong area of research out of the BER program. It's a strong area of research out of EMSL, where we're trying to understand how these microbes basically transfer electrons to these species, thereby immobilizing them in the subsurface environment. If you immobilize them, you know where they are. They're easier to accommodate and handle in terms of remediation from that point. So it's an example.
Make sure I understand. You've got microorganisms that take radioactive material and demobilize it.
Yeah, it's basically a redox reaction where they transfer an electron to the species and therefore change its oxidation state. And what happens is it goes from a soluble species, one that's soluble in water and therefore migrate, to an insoluble. It may precipitate into little nodules on the surface of these microbes, and therefore they don't migrate through the subsurface. So it's a really nice example of how biology is actually helping to remediate a natural example. The challenge is to understand that process so that you can perhaps engineer other processes to do similar types of things. So that's one example. Then there's another way in which you can use computational tools to actually simulate plumes and their migration and transfer through the vadose zone out into the subsurface environment and start to mimic the reactions along the way as they go. And so it brings together both experimental and computational resources.
Great. Next week we'll have a hearing in this committee on harmful algal blooms, which are a growing problem. We have great interest in ocean health issues. And any insights into that? I'm intrigued by, I know, Dr. Campbell, your lab's working on some things related to that. Dr. Patrinos in a different direction using algae. Any insights into that, which in the Pacific Northwest and around the country, this is a bipartisan, multi-regional, my partner in some of the work on this is Connie Mack, a Republican from Florida, and so you've got both ends of the corners of the country dealing with this. Any insights gained from your work or potential that you see? Just while I've got you here, we're going to have another panel next week, but I know you're doing some work on this.
Well, algae are among the most ubiquitous of species. I mean, they exist everywhere, in the marine world especially. And we are, over the last few years, through the power of genomics, understanding them more and more. Many of them, their genomes have been sequenced, specifically by the Joint Genome Institute. So insights into that biology, the biology of algae, can also give us the opportunity and the tools to fight them where they are not helping us. where they are hurting the environment primarily because of the insults that we cause, for example, through many of the fertilizers that end up in the Gulf of Mexico and cause the hypoxia, which generates the algae blooms.
So you feel like some of the insights you're gaining by just working on the genomics of algae could help us understand that?
Absolutely.
Dr. Campbell or Dr. Keasling, either one?
I might just mention that a lot of these algal blooms are due to pollution, as Dr. Patrinos said, that it goes into the ocean. And the way these are often cleaned up in municipal wastewater treatment plants is through microbiology. Mm-hmm. Actually, microbes accumulate the phosphates and other nutrients that end up in the ocean, or would have otherwise ended up in the ocean. Using some of the tools that BER has developed, the Joint Genome Institute is trying to understand those microbial communities. So they've sequenced the communities from these wastewater treatment reactors, and they now understand the microbes that are responsible for accumulating phosphates, for instance. And then this can help us design new wastewater treatment plants that are much more effective and lower cost at cleaning up these harmful chemicals.
That's a great example. Thanks. Dr. Ehlers, do you have any further questions or comments?
Just a comment. I appreciate this last interchange because I was one who wrote the legislation about the algae blooms, and it's becoming a problem even in the Great Lakes, much to everyone's surprise. So it's becoming more of a universal problem, and anything you can do to help solve that problem is helpful. But I also want to conclude just by thanking you. It's a terrible experience, frankly, to be a scientist in the Congress because you tend to starve. You know what the intellectual community is like, the research community, and how you're constantly interacting with people, generating ideas, and so forth. And there are very few scientists to talk to here. And so you've really enervated me again, and I just want to thank you for that.
Dr. Ehlers, thank you. I was negligent when I mentioned that my work with Connie Mack. Dr. Ehlers really has been the lead on harmful algal blooms for many, many years, and I've been privileged to work with him on that. He really, as in many, many cases, is seeing things that other folks aren't looking at. And so, Dr. Ehlers, thank you for raising that issue. You've been a champion on this issue many years. Starving intellectually in the Congress is an interesting observation. We'll just leave it at that. Any other final comments? No, this committee is sort of the brain.
Especially the subcommittee. This is very
committee.
intellectually stimulating.
And today was no exception. A fascinating element of research that I think many of us had not been fully apprised of before. I'm grateful for your service to the country and your work and to research, which is very, very exciting. And we look forward to working with you. And with that, I thank you for your time here and all our witnesses and the staff who put this meeting together. And hearing stands adjourned. Thank you very much.